The lactose present in the growth medium of bacteria is transported to the cell by the action of option A, permease. Now, a permease is a type of transport protein that facilitates the uptake of lactose into the bacterial cell. And in the lactoperon, in E. coli, the lactose permease encoded by LAC-Y gene transport lactose across the bacterial cell membrane into the cytoplasm. Thus, it is the correct option. Option B, polymerase. It is incorrect because it is an enzyme involved in the synthesis of DNA or RNA. Option C, beta-galactosidase. It is also incorrect because it is an enzyme that breaks down lactose into glucose and galactose. Option D, acetylase. It is also incorrect because it is an enzyme involved in acetylation reaction, that is adding an acetyl group to a molecule.